banana, and then followed by John Thomas. He is the expert of banana bunchy top virus. So nobody, I think everybody knows John as the expert of banana bunchy top virus in the world. And then actually we have another resource person, Dr. Lilia Carvalhais. Is she already on? No? Just want to say hi to Lilia. Lilia is the scientist in University of Queensland. She is supposed to come to Bogor, but unfortunately she could not make it. So Dr. Lilia will give the lecture from Queensland, from Brisbane, Queensland. Okay, and also for the guest lecture, we have some attendant in the online. Yeah. So how many already joined the 17 now? Yeah. Okay, I think I'm not going to give any more word, and it's better to start. But before we start the first lecture, I think we should take a group picture first because everybody is still fresh now. And we are all still gathered here. So please take a position over in front of those poster flyer.
First row, maybe.
Yeah, just sit. Yeah, and then the rest can stand up at the back. Okay, and then after pictures, we can start the. Yeah, you can also.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Thanks for coming in our plant pathogen diagnostic workshop. So today, as Professor Asti mentioned before, we are currently doing the guest lecture, which we have three resource persons. The first one, the lecture will be delivered by Professor Awang. He is from Department of Agronomy and Horticulture, IPB University. And the second one will be delivered by Associate Professor John Thomas from the University of Queensland. And the last speaker is Dr. Lilia Carvalhais. She is also from the University of Queensland. So thank you so much, John, Pak Awang, and Lilia. So Lilia will be delivered the lecture from Brisbane. Okay, because the time is already 9 p.m., so I will start the guest lecture now. Beforehand, I will start to read the short curriculum vitae from Professor Awang. So Professor Awang Maharijaya is a staff from IPB University. Currently, he is a director for Center for Tropical Horticulture Studies at IPB University. He also head of the International Study Program, Smart Agriculture, Faculty of Agriculture, IPB University. For your information, Smart Agriculture is a new study program at IPB. And he is also chair of the Geographical Indication Expert Team and head of Teaching Industry Faculty of Agriculture, IPB University, which is when we have a field trip tomorrow, Professor Awang is the head of the research station. Okay, so he graduated from Horticulture, IPB University, and Master of Agronomy, and in 2003 and 2007, and he graduated for his PhD in plant breeding and biotechnology, Wageningen University in 2013. Okay, Professor Awang, time is yours.
Because it's guest lecture. I took your friend. So it's a friend lecture. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. So thanks for the introduction. In this today and tomorrow, I think we will have more interaction, including in the fields. So I will talk a little bit about the current situation of banana in Indonesia in my capacity as the director of the Center for Tropical Horticulture Studies, because I also work with banana, but for now mostly on ornamental banana. Although now we are starting again for the BBTP and Pusadio resistance banana. Can I have my slide, please? Okay. I bring my own. What is it? I prepared them.
Oh, okay.
Always nice preparation. And this one is backup plan.
Be gentle.
Yes.
Be gentle.
Yes. Backup plan. Okay, so my presentation is about the banana status and diversity in Indonesia, and I want to emphasize on the for us to more understand about the potential and challenge of banana cultivation in Indonesia. Yeah, then after that, you will get more detailed information about one of the biggest challenge in our banana cultivation is the pest and disease infestation or attack. So tomorrow, I think we will visit this place. We have actually many variety of banana plants in the field, but I don't know if tomorrow all will be ready. But one thing for sure that I will show you some new banana ornamental plant that can be eaten as a food. And I will try to serve you all, but I don't think it will be enough. Let's see tomorrow. Okay. Next. Oh, and okay, okay. Like this, okay. So the outline of my presentation today is not too difficult to understand. I guess first the importance of banana in our situation, and then how about the economic potential, banana diversity that we have today, and challenge and opportunity for banana cultivation in Indonesia, and strategies for developing banana cultivation in Indonesia, and also some conclusion. Yeah, and we can discuss about the conclusion afterward. So banana is now becoming more and more important in our diet. We have almost 6% increase from last year to from last 2 years to last year's production and also consumption, because everybody now start understand about the importance of good diet, right? So everybody want to eat real food, not junk food. Yeah? Every morning, my wife, don't eat this, don't eat that. They prepare because now you look so big, yeah, and then okay, I to consume more fruits in our situation. Yeah. So everybody looking for banana and next, yeah. And many things can be also produced from banana. Also, I always say to the governments that this not come from one type of banana. So when somebody from from government, they say that banana is multifunction, it can be produced for food, for textile, everything. Yeah, they should know that this is not come from one type of banana. So don't mix it everything in one. So if you want to, if we want to do for food, we should breed the type, certain type of banana from for the textile and craft. We should also provide different type of banana. I forget to use textile batiks from fiber of banana. Yeah, this is in Kalimantan. It's very popular. Okay? If you visit there, for instance, in the South Kalimantan, yeah, anybody from there? Rida, yeah. So you can buy in the shop. Yeah, and then for vegetable and I don't know. There is a very debated issue about tomato. Is it fruit or vegetable? But actually, for Indonesia situation, also we should debate banana is fruit or vegetable, because the use of banana sometimes the raw material of fruits also we put it in the kind of traditional salad type of dishes like rujak cingur, where we use unmature fruit banana. Next. Yeah, and banana in Indonesia is almost 35% of fruit production nowadays in our situation, and it's predicted to be increased next year, because there are many industry now become expanding their business. For instance, in Sukabumi, they also, I don't know, start doing something there, right? And also in Blitar, Jawa Timur, they have a huge banana plantation. Yeah, next. And this is to add information that now you can also buy single banana in the convenience store, such as Indomaret or Farmers. Yeah. In previous 3 years, this is not trend, but now it's becoming trend that banana also treated as a snack, yeah, in our society. So you can see here the production of banana in Indonesia is increased now, up to 9 million tons per year, and also the consumption of the people in Indonesia has increased. And where are the next? The rest are exported to many countries around Indonesia. Yeah, so this is the increase almost 6%. Yeah, so it's very good business they made. Yeah. Okay, next. Yeah. So this is just to give you information how the banana developed in Indonesia. So last year, maybe in our mind, we have a image that Indonesia is the fourth biggest producer. Yeah, but now we are at the third. This is the development of the growth of banana plantation in Indonesia. Ecuador, India, Brazil. Now India is number one. Oh, China, sorry, China. Where is China? This is still, oh, 1977, so coffee break, no? No. So we can skip this, right? Yeah. So we now do the next slide. Yeah. Now we are in the number third. Applause. No? Applause. We're in the third biggest producer of banana. Yeah. Okay. See? Next. Yeah. And in Southeast Asia, we are competing with Philippines, of course. But Philippines is more strategic because it's closest to China for export. Yeah. To Indonesia to China, maybe 4 to 6 hours by flight, and from Philippines only 2 hours. Yeah. So yeah, that's not the advantage of us. So we have to produce cheaper banana, yeah, with good price to go to China. Next. Yeah, and the situation now, banana cultivation is primarily still done by smallholder farmers. And the good thing is that now they have a collaboration with big traders, and they start to apply standards, etc. And in the smallholder farmers, it's more, I think it's better because they plant ambon, barangan, pokok, tanduk, and many kind of plants. Well, recent years, there are large banana planting business, yeah, and they start to plant a huge area of plantation of banana. And this is in Subang, yeah, and also in Blitar, East Java, yeah, they produce a lot of banana plantation, yeah. But yeah, this is very logic that they plant all Afghanistan of banana, and then as agriculturists and also pathologists, we know that this is very risky to the damage by the pathogen. Well, next, yeah, and East Java is the biggest producer in Indonesia. Yeah. 2020. The good, the new data now, the students, and then this one is growing. Next. Okay, what type of banana that present in Indonesia? I think we have a hybrid between mostly Musa acuminata and Musa balbisiana, right? And not, I think all of them are still can be grown in Indonesia and still produced by the farmers. Therefore, we have a diversity of banana for consumption and also for ornamental, and I mean the concentration of banana in Indonesia is unique because the wild type even people still can use it for many purposes, such as for textile or exporting the leaf of banana. Yeah. So do not expect export leaf from Cavendish type because the quality of the leaf are not good. So we have to plant pisang batu, banana, Prof banana, to get good quality of leaves for export to Europe, for example. Yeah. But the rock banana or pisang batu is not good because it contains seed. Yeah. So it's not very pleasant to eat daily. Yeah. Or you lose your teeth. Okay, and in Indonesia, this is happened because there are many use of banana for cooking prosperity. Yeah. So sometimes people do not eat it when it's too ripe, so they get the unripened banana for making a sauce or something like that. Yeah. And then they use it for fried, boiled, or steamed banana. And also there is a regional variation. There is a local agent that it can only produce in a good quality in certain area. Yeah? And this is very good because it relates to the indigenous or traditional dishes that they eat daily. So that's also a form of conservation in our situation, right? And also medical stage. Okay, next. This is a sample of local Indonesian banana varieties. Raja Bulu, this is a very good price actually. Yeah. So if you buy in not Sukamati, it's very cheap, yeah, of course. The producer, tempat bibisan yang mahal tuh. Oh, bukan Opres. I'm sorry, I forgot the brand, the name. Pasar apa itu? Sukasari, ya, Sukasari. Sukasari. Sukasari, ya. Kalau kita ke pasar Sukasari, if we go to Sukasari market, the Raja Bulu banana is very expensive. So one cluster is can be cost more than 150,000 rupiah. So it's very expensive. But in Sukamantri, we sold it maybe 90,000, so half price. Yeah. And many kind of one, and even the smallest one, like pisang reja, is a very major banana. It's very popular usually for ceremony, because in the ceremony, like the wedding ceremony, then people come very often and many invited, so they can eat it more and more instead of having one big and then they small like a snack or something. Yeah. Next. This is the example of local legend. Pisang kepok Nias. Anybody from Nias? No? Yeah, Nias is in the North Sumatra, right? And from North Sumatra, you landed in the airport, and then you maybe spend half day or more than half day to reach Nias using a boat there. And sometimes you cannot cross because the way is too big. Yeah? They have a very good quality of kepok type, and this is a very a good taste according to them. Why according to them? Because as I mentioned, this is a local, so maybe for other people it's not a good one, but there is a consumer for this, and demand is also high. So we do the geographical indication branding for this banana. Why? Because we want to respect this banana. Yeah. So by branding of the using GI, GI is a geographical indication. Usually now is very familiar for coffee. You know, For coffee, you can have a coffee gayo, Arabica gayo. You can get the coffee Java, etc. So for banana, we start to think about the same thing because in the South Sulawesi, for example, for pisang keke, yeah, it is another very unique. Yeah? So very unique. The taste is also very unique, and banana is not from other area. They have a local legend banana. Yeah? They only want to start branding this, so they will get a higher price. So it's a good way for conservation, right? So instead of only preserving, only collecting, and only put it in the facility of like in a preservation area, then we start to use it as a program, right? Okay. And it can be exported also in Vietnam and other north side of Nias, yeah. And it has economic potential and also can be used as a food security for people in this certain area. And it has a nutritional value that can enrich the nutrition status of the people living there. Yeah? Instead of eating rice, it's good for banana. Next. And then the challenge and opportunities for banana cultivation in Indonesia. Yeah, is the something that you have to discuss intensively today and tomorrow. Yeah, the pest disease. Yeah, even a report in Wageningen, they say that this is perhaps the most endangered fruit. Yeah, because if everybody plant the Cavendish stuff everywhere, yeah, and there's an outbreak. For instance, now it's Fusarium and PBTV virus, then we are in danger because many people now already depend on banana. Right? You cannot replace banana with mango or something. Yeah. And then the second one is the ornamental. Next, ornamental banana is now very popular in sustainability. Yeah? Instead of only this type of ornamental banana and this type pisang kipas. Yeah? Now they also develop something like this that we can buy in the nursery. Yeah? This is maybe 1 million rupiah. Yeah, good price, right? This is very good price, and it can produce this type of banana fruits. Yeah, and it can be eaten safely. I ate several times, and I'm still alive. So this is a very good one. So we can try to modify it. Next. And then the third, the cultivation technology that can be improved in our situation. Yeah. First is tissue culture. Now we develop and push the seed producer to use at least tissue culture to get the disease-free material instead of only from the corms. Because the seed from corms of banana is cost 5,000 to 6,000 rupiah. But for tissue culture, now it's become cheaper and cheaper. I checked tomorrow, I checked yesterday from Java, and they sell it to me. I hope it also can sell to you the same price. Yeah? It sell to me 1,000 million per plant. But you got disease-free material. Yeah, when you use corn from the field, then you don't know what is the status of the pathogen side. Yeah. So for 4,000 more, it's the gap is 4,000. So maybe it can be less and less. So we push the research to get the disease-free material, but with affordable price. I think it's if only like that 5,000 compared to second thousand, maybe they will start to use tissue culture material for planting banana. Yeah. And then also we try to develop disease-free, disease-resistant. So this is different, right? This is disease-free material, but the second one is disease-resistant plant. So we should start continuing breeding for resistance. Yeah? Because this is needed for integrated pest management. Yeah? If you have a disease-resistant variety, then your jobs using another approach in integrated pest management is become less. And this is very important. Why? Because we can reduce the production cost. If we just like now, and we cannot compare with Philippines, Vietnam, maybe one day we cannot compete with Vietnam, because they have a closer to the export market compared to us. Yeah, so that's something that we should solve together. And developing monitoring system, precision irrigation, because now the availability of water resource is also a concern in agriculture. Therefore, we develop a smart approach and technology, and we start to get the critical mass of smart agriculture people by developing the new study program, smart agriculture, because it's really needed. We have to start thinking that we are not competing to produce higher and higher production only, but we should also lower and lower the production cost. Yeah? Fertilizer optimization and identity determining system, improve storage facility, and ethylene management for export. Okay? Next, just give you an idea that the China market now still very wide open for us. Yeah. So this is the opportunity for us. Yeah? So we have to produce with less input to get a good price, but still high quality. So we have competitive products, and it can help us to enter the new market. Yeah? 10 years ago, somebody comes, yeah, and they want to export the banana to Saudi Arabia. But in the end, they just decide, no, I want to sell it in Indonesia. Why? Because the price of banana in our situation is good. It's better than the price in Saudi Arabia. Yeah. Therefore, they say, okay, we can sell it to Indonesia. Yeah, they got profit to it. Yeah. Next. Okay, and the government policy. I can tell you that everybody now is working with banana. Yeah? There are many programs in government, such as Indonesian Central Bank, to develop 5,000 hectares of new cultivation of banana back in this time. Yeah, in South Sulawesi. And they already start thinking to replicate before starting. Before, I think there will be subsidized an incentive for banana cultivation. So we need research and development for that to give. I think everybody can be consultants to the program. Yeah. IPB alone is not. We have a limited resource there. Therefore, I mean resource in human resource to guide the governments and also the Central Bank of Indonesia for developing this program. So I hope everybody that join this workshop can be recommended. Yeah? If somebody asks me in this location, then I can ask who's the expert. We can sell it. So we really need to do the research and development. Is it right to make a cultivation there? But mostly sometimes they still eager to do it, right? So we should think the next one, how we can minimize the bad impact, the risk. Because sometimes we cannot say, don't do that. We cannot say that to the government sometimes because they have a target, they want to do the program. So what we can do as a scientist, of course, we can say or give them some notes about the potential something that can be bad or what that we can prepare first to face the challenge. And then improve market access and export promotion. The government now always asks the producer of banana to promote their products in many countries, and they start to build infrastructure development. And now we also developing the quality standard and certification, RSNE standard. We develop because we have a non-user tariff, right? So we have to develop standard for the banana in our situation and also for import. And then supporting smallholder farmers using extension service and training and microfinance and credit schemes for banana. Next. This is the example. This is from Central Bank of Indonesia. They for developing banana cultivation, this is based on our discussion. First, is the resource and factors of production, and then we should take a look at upstream and downstream industrial development, and also development of infrastructure, human resources, institution, and supporting factors, research and technology development, and development of financing aspect. And this is the example next. It's too small, but I think everybody will get this copy of presentation. You can see this is the example of its strategy in its deadline. For instance, this is human resource and production factors. The strategy number 1 is to implement program for community development of banana, and then list all the farmers, and give a farmer's card, and then we can do it after. Next. So conclusion, banana again is a vital food crop with significant economic potential in Indonesia, as you can see. Each year they increase the production, so it's good business, it's good money for producer and for consumer. They can get benefit from that, availability of high quality products to enrich their nutritional status. And diversity, we are lucky because Indonesia has a high diversity of banana, and the good thing is that industry still going with apprentices, okay, smallholder farmers, and society is still eager to eat many types of banana, and some of them are having a good price, even higher than apprentice itself. And local agent branding already started, but we have a challenge and opportunities. So we should do many of things together, and therefore we should keep this society, keep this consortium, and strategies. We can discuss about comprehensive strategies that are needed, and but although it's a comprehensive, maybe it's also local specific strategy for its development. Next. I think that's all for starting the discussion today. This is just a quick view and thinking by me. Maybe somebody can add many new things. I'm really welcome. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you so much, Professor Awang, for very great lectures, and I think it's very interesting regarding the variety and opportunity and the most importance of the banana in Indonesia. John, sorry, because Professor Awang will have an occasion after this one, so we will start for the first term of discussion. So everyone in this room or participants through online, please, if you have any question, please raise your hands or type the question, or you can ask directly through Zoom. Thank you.
Langsung aja Pak. I can hear you.
Thank you. I would like to question for the last slide. Actually, local lead. I think
Cukup.
I was I'm from sorry,
I'm talking.
from other university. But it's a very different. How to bring the local native like oh, say Sulawesi, so Sulawesi government has program to
Plan many campaign this Friday. I think you can.
maybe Bacterial government, but he's not government. So how to bring the local religion
Compare the
appendix.
local brand like Kebab,
Or not interesting. The local legend not interested from the government. Also
branding the local legend. Actually, Kebab variety. I think. Okay. Thank you for the question. So the question, if I'm not mistaken, is how we can start and how we can do the branding for a local legend like this. Yeah. So we should first really know about the reputation. Yeah. We cannot create suddenly something like this. Yeah. They call it local legend because they have reputation. Right? So for instance, Tandu in Bogor is really famous. Yeah. So Tandu is also and Raja Bulu is also kind of local legend that it has always has a demand from society. But the demand from society is less and less because the new generation do not respect this kepok. I mean, for Bogor situation is Tandu and Raja Bulu, right? Young generation maybe they do not know Raja Bulu, yeah. But if they try Raja Bulu one time, but they can see that there are many different experience comparing eating cafe this time. Yeah. So that spirit that we should manage. Therefore, we need to promote the good thing about the local legend. Okay. Yeah. For instance, also for South Sulawesi, maybe we can start from EP South Sulawesi that are not present in other place, right? So EP Sulawesi Selatan should be brand as a local legend in that area.
but it's Sulawesi too.
Yeah, in Sulawesi. Yeah. So after that, we talk to the government because this GI branding should include the government act. Yeah. They should give a recommendation that this is a really local legend that should be developed as a GI program. Yeah, and then after that, from the academician, we should explore what is the good thing about this in scientific way. Yeah, because for GI branding, we should know about reputation, quality, and also the geographical factors that contribute to the product. And the geographical factors is not only about soil, environments, etc., but also the people, technology, yeah, that they use daily to produce this product. Yeah, by that, this is very good for conservation. Yeah, because if this is treated same price as a regular coupon, then there's no use for people to conserve this, right? But this is very good because in the field we can see that it has good trees, and then this is resistance to several banana diseases. That's my answer. So we need to talk to the local governments that you have a localism that should be conserved. Yeah, but the conservation is not only by storage, but we can use it for a good products with higher price. That's my question. My answer. Yeah. Okay.
What is the opinion of local Indonesian people about Cavendish bananas compared to local cultivars? How well do they like the area?
The perception of Cavendish compared to the local banana. The Cavendish is more, more good in skin. That's all. I think that's the we have a research about that. Yeah, if I'm not mistaken, I'm not remember by heart. It's about 76% attribute that influence the people to eat banana is the skin, and that's the problem. Skin of banana. So maybe you should think about skincare. Modify the banana. Yeah. So think more into that. This really contributes to the unwanted preference.
So it's the appearance?
The appearance, the skin of banana, yeah. And can we come back to the Cavendish in the Indomaret? Back. This one. In our situation, yeah, in our field, because we also have contract with Indomaret at that time, we really push this because they reject every defect of it like this. Because they say I cannot sell Cavendish with skin like that. So we should maintain this nice skin. That's the problem. Yeah. So
that's what our producers say in
no.
the major area. They say they grow banana skins.
Oh.
They grow banana skins.
Yeah, that's the problem. So when already infected with something that destroy the skin, and cosmetic effect that is not good anymore for Cavendish. But for other local type of banana, they say okay. Even for tandu, when the skin become blackened, yeah, and they say, this is nice, but this will be sweetest. But actually not, yeah. It's different. Yeah. Okay, that's the perception of the consumer that we should also take a look into. Thank you.
So we have another question through the Zoom from Nurfianto Biarpanidu.
I will try to cover this.
Oh, okay.
Yeah.
Okay, for someone who.
I will follow maybe 10 minutes, and then I'll take a look at the question in the Zoom.
Okay.
That's fair. I want to give an opportunity for people here who want to discuss with me.
Okay, by Amy, please. Thank you for the opportunity. Thank you for Awang. Sorry. My name is Henny. I'm from Nusa Tenggara University. So actually, if you don't mind, I will have three different questions.
Okay.
The first one is also related with the one that we as mentioned.
kebab because in case of kebab, we consider now it's really so some people who want diseases or the bacterial blood disease. And then in our area, it's already wiped out almost all the kebab varieties. I do not know whether this is also resistant to blood disease bacteria. And then you were also mentioning one of your technology that can be implemented is using the disease-resistant varieties. I was wondering whether you are having already like places or alternatives for replacing that kebab if they were affected by the blood disease. And then the second one, you were also mentioning that there's one technology is using the integrated pest management and also monitoring system. Does IPB has already have that system that can be applied in another place? And also, yeah, and also then you were using the high density planting system, which is for us is a bit worrying because when we have the monoculture situation and then also the disease probably is going to be very heavily spread. In our place, we also have like local banana, it's called pisang luan, but I do not know whether it is deployed or deployed or whatever, but it would be possible becoming one of the air blossom that can be here.
Okay.
Thank you very much.
Thank you. So for the first one, you have a couple of times, but now it's wiped out by the blood disease. I read some information from Unti Sayang variety, for instance. Unti Sayang is from Kalimantan. After the second, you can check Unti Sayang. No, this one is the name of variety. About Unti Sayang. It has a resistance to blood disease because the flowers are not developed again after processing the bunch of the fruit, so the infestation of the blood disease will be reduced. I think you can try that first, yeah. But I don't know the latest development of that variety. The quality of the fruits is good. Yeah, we have that collection in our field station in Tanjung. But I have no experience recently to take a look for its resistance. But it's still there, so maybe it's resistant because we also had a collection of banana gem plants in the field, and the Unti Sayang cluster are still grow well at Tanjung. Maybe it has some degree of resistance. I don't know. Yeah. You can also try to do rotation breeding using the pot that you have in the location. Maybe you will get something. Yeah. Also, if somebody, I don't know, the blood disease, and who actually can talk more about that, I don't know. Is the gene for blood disease already characterized or something? Yeah. If that's the case, then you can do mutation breeding with targeted, such as genome editing for that situation. Monitoring system. Okay, the monitoring system has developed by some companies in Indonesia for the banana, but I could not share the picture because they did not allow me to take a picture of the system, but it's already there. This kind of using a drone and also a picture from about the area of the trees to monitoring the development of Rosario and also Bacterial, because there will be a spot of different color, right? So I think that's easy, but I'm not trying by myself. Now I'm trying with the virus to see if also works with that. But until 5 meter 4 meter, 2 meter, they cannot really distinguish between the virus and others, yellow caused by other factors. So maybe for banana it will be different. You can check or you can visit to some company for banana. Maybe they can sell it.
You said that is what is the
Sorry.
company that you
Now many companies now start working. So it's not only DTP and many companies now they start doing something with banana. In Indonesia, now, the development of agriculture is very good. Yeah, I can say. For instance, if you travel along the highway, you can see banana plantation in right and left side and right side, and also you can see a huge greenhouse area. Yeah, so many company now is working on that. Yeah, actually, yeah. The last one is
high density.
high density. Yeah, that's also something that we should really take and do some adjustment on it. Some company they say it work well, even some farmers because they have very limited land, they use high density and they produce it very well. And I can imagine because they only work in 3,500 square meters, maybe it works. But if you do it in many hectares, 100 to 100 hectares, maybe you will get a problem with that. Yeah. But for small holders, farmers with very limited land, maybe this high density crop for banana will be very good. Yeah. Okay. Thank you. Still there.
Probably one more question.
One more question. Student.
Students.
I will provide you a box of chip tomorrow.
Yeah.
Potato chip, sorry, potato. Okay. Yeah, variegated banana is so expensive because the number now is still limited. I think that's the main factor. And because it's variegated, everybody now loves variegated, tomorrow you can see a plant with very high price. So we put we put attention on this because this is very good, but still limited. And the good thing, very good for business, good and very relative. This is very difficult to propagate using input. That's the good thing for that's the good thing for producer. But yeah, okay, that's maybe that's the main contributing factor for it. And if you have a wedding ceremony, you serve this, everybody call you Sultan. Sultan is a king, because you have a lot of money there. Okay. I will answer the question in the Zoom using typing after this. Yeah, I will stay there maybe after. Yeah, then we will do this. Thank you. Thank you.
Thank you, Pak Awang, for very great lecture. Please give applause. Okay.
So everybody will get a tip. Okay.
Okay. Thank you, Pak Awang. Okay, we will move to the next speaker, but before John starts to deliver his lecture, I will start to read his CV. Okay. As Associate Professor John Thomas. John Thomas has worked in plant virology over 50 years. Can you imagine? He has already had a very many experiences. So covering virus and phytoplasma diseases of a wide range of crops, including vegetables and tropical fruits. grains, bulbs, and ornamentals. So he obtained his PhD from the University of Sydney on tobacco yellow dwarf and bean summer death viruses, working firstly in the Queensland Department of Agriculture. And before he retired, he works in the Coffee, Queensland Alliance for Agriculture and Food Innovation, and he is also my supervisor at UQ. And a recent major project he led has been funded by the Bill and Melinda Gates Foundation, BBTV mitigation, community management in Nigeria, and screening wild banana progenitors for resistance. Now he is based in Brisbane, Australia, has recently retired, and I'm the last his students. And now holds an honorary associate professorship with the University of Queensland. Okay, Jen, time is yours, and I will share your screen.
Can you stand up here?
Oh, yeah.
Because the camera is over there.
Ah, right,
Just
right.
right here.
Oh, right.
Sorry.
Selamat pagi. Maaf, saya tidak bisa berbahasa Indonesia. Saya bisa perlahan-lahan
Okay.
dalam bahasa Inggris. So, in English and slowly for you. I'm giving this talk. Kathy Crew unfortunately was unable to attend, as Peter mentioned. So I'm using some of the information that she prepared for another workshop we gave in Papua New Guinea recently. So this talk is my work and Kathy Crew's work. Next slide. So you know who we're talking about. This is Kathy Crew, and this is Lilia, who will give the next talk. This was taken in Papua New Guinea recently. And on the other side, I've visited Indonesia many, many times now. I think it's something like 20.
Yeah.
And worked with Asti especially, and Ita many, many times for many years. And this was a photo with Chacha as well. Yeah. Taken in Sumatra last year. I was working on chilies in that case, worked on bananas and surveys and lots of other things. But that's just to show you who should have been here if they were able to, Kathy and Lilia. Okay, next. Right, I'm going to talk about banana bunchy top virus, and I think most people haven't got a detailed knowledge of virology. So it'll be a reasonably simple talk, but if you want to ask any more detailed questions, ask when you want to. Banana bunchy top is a very, very serious virus disease of bananas. And this is the sort of devastation that can occur. This is a plantation in Australia a couple of decades ago now, but when control was lost at the Cavendish plantation, wiped out by the virus. Okay, next slide. Right, the symptoms. We haven't got the point over. Borrow yours. Work very well. Yeah. Give it back. Right. Banana bunchy top has very characteristic symptoms in some cultivars. In other banana cultivars, you don't have all the symptoms, or you may have very few symptoms. But I think all of these are in Cavendish. and Cavendish bananas really show severe symptoms. Plants can be stunted. They have yellow leaf margins, the edges of the leaf, and they tend to curl upwards. If they're not flat, they curl a little bit. If you look closely at the midribs and petioles, you get dark green streaks. And in some cultivars like Cavendish, you get these little dots and dashes on the leaf. You can see those when you hold the leaf up to the light. And not look down on it, but look underneath it. Suckers, young suckers can be very badly affected. The main reason the virus is a problem is you generally don't get a bunch of the infected plant. So once the plant's infected, generally not a bunch. If it's infected very late in the cropping cycle, you might get a bunch, but they're often small, distorted bunches. And if it's infected very, very late, sometimes the only symptoms you'll get on the plant are these dark green streaks on the tips of the bracts, because the virus is infected too late to show symptoms in the leaves. So that might be the only symptom you see in a recently infected plant that's about to produce a bunch. But in the next cycle, you almost certainly won't get any bunches. Okay, thanks. Right, and to give you an idea how quickly the virus can get out of control, this is a plantation where we were doing some epidemiological work, and the farmer stopped controlling the virus, and now almost all the plants are infected. In a period of 9 months after stopping controlling the virus, gone from a few percent, 5% or something, up to almost all the plants. And this is another example where there are gaps in the plantation where infected plants were removed, but the farmer couldn't control the virus, and 18 months later, there's no bananas left that weren't infected. And this is a distorted bunch from that plantation. Okay, that's fun. Right, so where does the disease occur? In most places where bananas are grown, in the tropics and the subtropics, with the big exception of South America. So it occurs widely in Africa, South Asia, Southeast Asia, in the Pacific Islands. In Australia, it only occurs in one small area, in our subtropical production area. And about 95% of our bananas are grown in the tropical north, up here where we don't have bunchy top. So that's a major quarantine threat for the Australian industry. In Indonesia, it occurs quite widely, different incidents, but I think there's a spot on there for most of the islands that people have come from here. And the reason there are two different coloured dots is that there are two basic groups of strains of banana bunchy top virus when you look at their genomes. But the symptoms are the same, the effects on the plants are the same, they're spread the same way. So for our purposes, it's all the one thing. Okay, next. So how does the virus spread? Firstly, the virus has, these are virus particles. There. These are the virus particles. We call them isometric. They're almost spherical. They're only 20 nanometers in diameter. So they're actually the smallest viruses that are known for any organism. Nothing any smaller in animals or plants or bacteria. They're spread in nature by aphids. And it's called circulating propagating, non-propagating transmission, which means the virus travels right through the body of the aphid, takes the virus up in its mouth parts, goes through its whole body, and then gets re-injected with its stylus, so it circulates. Aphids have two major forms. Mostly you see wingless aphids. When they get very crowded or when the plant's dying off, they produce a cycle of aphids with wings, which can fly. And it's the aphids with wings that are more important in spreading the disease. It's a long way for an aphid to walk between a banana plant, much quicker if it can fly. Okay, so the virus is spread by aphids, and it's also spread in planting material. And it doesn't matter what sort of planting material, if it's tissue culture or suckers or corms, the virus is transmitted in all those forms of planting material. So tissue culture is another form of vegetative propagation. If you try very hard and use meristem tip culture, you can eliminate the virus. But if you take big pieces of tissue in tissue culture, it's transmitted just like any other form of vegetative propagation. Just because you've got tissue cultures doesn't mean you've got virus-free plants. So they're the two ways it's spread. Locally, mostly by aphids. Long distances between different regions, mostly people carrying infected planting material. So if it's moved between different islands in Indonesia, it's mostly people carrying planting material. Once it arrives, it can spread via aphids because the aphids occur everywhere. So even if you haven't got the virus, you'll have the banana aphids on bananas. Okay, next one. This is just a couple of shots to see what the aphids look like. Sometimes people don't realise they've got them because they're quite secretive aphids. They like to hide under the leaf sheets or around the base of the plant, sometimes even under the soil. So often you don't see them until you peel back the leaves and have a look. But they can occur in very high numbers, hundreds and hundreds on the young growth. Okay, next one. Yeah. Just another couple of shots of aphids. There are just a couple of shots we took in New Guinea when people didn't think they had aphids. So we peeled them back and showed them where the aphids were. Okay, so. And for the next two slides, we've seen Buasi and Bernama. This is some of the very, very good work they've done in our Gates project over the last few years. I said the virus is transmitted by the banana aphid, it's Pentalonia nigronervosa. We now know it's also transmitted by the cardamom aphid, which is a related species. It's Pentalonia calladii. Now, Pentalonia nigronervosa, the banana aphid, mostly is found on bananas. About 95% of the black aphids you'll see on bananas will be nigronervosa, but just occasionally you see Pentalonia calladii on bananas. The other way around, the Pentalonia calladii likes to live on a lot of other plants, especially in the ginger family. So 95% of the plants on alphinias and gingers and turmeric and so on will be calladii, but just occasionally you see nigronervosa. And yeah, with nigronervosa on bananas, for instance, the aphids seem to like bananas with more B genome and a more Galbiciana genome than a Cuminata for some reason. So you need to keep track of what Bernama is doing. It's very interesting work trying to work out why that happens. We have no idea yet. Okay. Second slide is just to show that both species will transmit banana bunchy virus. These are the portions of infection. But with the work here in Bulgur, the nigronervosa was more efficient at transmitting. But nevertheless, the other species also transmits the virus. Okay. Right. That's okay. It's fine. Just quickly tell you how we control banana bunchy top virus. it seems that all edible bananas are susceptible. They differ a lot in how badly affected they are, and Cavendish bananas and pisang mus, for instance, are very badly affected by banana bunchy top virus. Other cultivars, Tunduk might be an example, and Sabah, Philippines, don't get infected as often, but when they are infected, they show symptoms like what other cultivars. So different cultivars of banana vary in how easily they are infected, but they're all susceptible to some degree or other. So we've got no immune varieties of bananas that you can eat. So there's no good resistance in that case. So the only ways you can control it are to eradicate infected plants and then replace them with healthy planting material. And at the same time, you use quarantine. So you carefully don't introduce infected planting material into the new plantation, and you carefully don't take planting material that's infected out of an affected area. So that's in Australia, the industry was almost wiped out about 100 years ago now, in the 1920s. Something like 90 to 95% of bananas were wiped out with banana bunchy top virus. And they worked out this control strategy, and that's what's still used today in Australia. And it's taken the incidence from 95% down to about 0.1% now. So it still occurs, can't eradicate it, but it's very, very rare now in Australia. Just occasionally there'll be a plantation where it gets out of control. But if we want to work with it, we need to talk to the people who do the inspecting and find out where the infected plants are, because we can't find them ourselves. There's just not enough of them. So it's very old-fashioned, but it works. It'd be much nicer if we had some resistance. And there's a lot of work going on looking for that now. In our Gates project, we found that in the Philippines, Musa balbisiana, the wild Musa balbisiana from the Philippines, many accessions of that seem to be immune to the virus. But that's a wild seeded banana, full of seeds, like Professor Awang was talking about. Seeded banana, can't eat them, but it's material that you can start breeding with. Okay. Right, and the top row is how the control was done in the old days. You would spray the plant with something like diesel or kerosene, which kills the aphids, because you don't want to shake the aphid up if it's on an infected plant and then make it fly away to the next plant. You want to kill it on the plant that's infected. Then you dig it up and chop it into little pieces this big that won't grow. Very, very hard work, but that's what was done 100 years ago in Australia to save the industry. Now they spray with mineral oil and inject the plants with herbicide and insecticide for plants. And all the commercial bananas in Australia are inspected regularly in the bunch club areas to control the disease. So that's the old-fashioned still control method we use. Yes, for a long time, banana bunchy top was thought to only infect banana or plants in the Musa genus. And over the last maybe 20 years or so now, we've found that there are many other alternative hosts. Alternative hosts of plants that can also be infected. But quite interestingly, they're all hosts those two aphid species will feed on. The aphids need to be able to feed for a long time on infected plants to be able to take the virus up. So it's got to be a host that the aphid likes to feed on. But quite interestingly, There have been many tests for alternative hosts. And many of the tests have been done very well, and we're sure that the result is correct. People have been able to infect the plant. Sometimes it shows symptoms. They've been able to detect the virus by PCR or ELISA in the plants, but we're sure that it's true. Then another laboratory tries to do it, they can't do it. We did a lot of work maybe 30 years ago, trying to infect a whole range of alternative hosts, and 0% in everything. And 100% in all the controls, positive controls. We use the same varieties of plants, same species, same variety of plants that other people use, and they got infection. So what I have here is examples of plants like canna. Number of people have found it's been infected. But been two separate lots of experiments done in Australia, can't infect canna in Australia. No, lots of them. Unfortunately, I see my own name there in all the failures. But here's one. Kerma, let's go on. Oh, ginger, yeah, not ginger. In Indonesia, infected. In the Philippines, can't infected. And both tests done very well. No problem. So what we think is that people in the past didn't differentiate between the two aphid species. They all look the same. If you just look at them, they didn't know whether they were working with Pentalonia calladii or Pentalonia nigra. So they might feed differently on the hosts. I mentioned there are two main strain groups of the virus. So they might be genomically a little bit different. And the hosts might be just a little bit different. So there's a lot of variables there. But what it means is there are many alternative hosts, but that doesn't mean in every little situation in your own production area, all those plants will be susceptible. So a lot of questions there, but there are alternative hosts. Right, so that's some of the biological information we've done. This is the computer model of what the virus particles look like, and we look very closely at them. Blow these up. The virus it's a DNA virus, so its genome is DNA. It has 6 different components of DNA in it. The DNA is a little circle in every case. So every gene is on a single circle, and every gene is encapsulated in one particle. So if you want a complete banana bunchy top virus, you need 6 different particles. So that seems very inefficient. The aphid's got to take up all 6 particles and inject all 6 particles, or you don't get an infection. But it works. Don't know why the virus does that, why it makes particles so small, doesn't put them all together. But not for us to say whether it's good or bad. It's a very serious disease that spreads well. So, yeah, that's what it looks like. 6 different genes. We know what all of them do except one component. We still don't know what that gene product does in the infection cycle. So that's what the virus looks like. Next slide. Many different ways of detecting the virus. In this workshop, we'll be just looking at one method. If we go back a step, the very first way you identify the virus is to look at it, look at the symptoms. And 100 years ago, that's all they had. And they managed to control the virus just by looking at the symptoms. People think you need the latest, best, biggest test before you can do virus identifications. Not necessary all the time. Sometimes just looking at symptoms is very, very good. but there are a lot of molecular methods now to detect the virus. The molecular methods either look at the DNA, so DNA detection methods, or you look at the coat protein of the virus, the outside that protects the virus particle. The proteins, they can use protein detection methods like ELISAs or rapid antigen tests. So there are lots of PCR tests available, and we'll be doing PCR tests in this workshop. With LAMP assays with mediated isothermal amplifications, recombinase polymerase assays, or detecting the genome of the virus. That's enough there. So that was the DNA. You can detect the coat protein of the virus as well. Do ELISA assays, colorimetric test. And this is just an example of some screening we did in the laboratory where the height of the bar is how much, it's related to how much virus is present by ELISA. The negatives are along the bottom here. And lots of colour development, high bars for ELISA. In our Gates project, we made some monoclonal antibodies and polyclonal antibodies to the virus, which are used to develop the ELISA test, but we also developed a lateral flow assay, like the rapid antigen test used for COVID. Exactly the same, it's exactly the same method. And that worked really well. We could do that in the back of the car in the banana field. And we got the results here in about 5 minutes. So this is the control line. It's like a COVID test, shows that the test is working. These are positive samples. These are negative samples. And when that test's working nicely, this is all the equipment you need. Just a little bag or something to crush the leaf sample up in, put the dipstick in, and wait a few minutes to get the result. Okay, right, so I said we're going to be doing PCR in this workshop. There's many different primer pairs available. Anybody can design primer pairs, but it's important that you use a wide range of isolates so that you know that you detect all the different isolates that might occur. Now, this is an old set of primers that were published in 1995, but they're still very, very good. We don't know of any banana bunchy top isolates they don't work with. It always work. The PCR tests target the DNA, so they target one of those 6 molecules. And we've chosen in this work, it's the replication initiation protein. So that's an absolutely essential component. Sometimes you'll be missing 1 or 2 components in isolates. We don't know how the virus manages to live without them, but it does. But they all need the DNA out of the web. So that's a safe one to use. They work with both subgroups of the virus. And very importantly, they don't cross-react with other isolates that are related, other viruses that are related. Yep, and you can hopefully see that on the first page. How much time have we got here?
So 15 minutes left.
Right, okay. Right, so that was fairly superficial. And you can ask questions if you want to know any more about any detail. But I thought I'd just show you that there are more viruses in banana than banana bunchy top virus alone. These are some of the symptoms. There's a virus called Abaca bunchy top, which Abaca is Musa textilis, grown especially in the Philippines for fibre. It's related but distinctly different from banana bunchy top virus, but it's spread by the same agent. It causes the same symptoms. And people didn't even know it existed because it caused the same symptoms, transmitted by the same agent. And some serology tests with some antibodies will cross-react. So you've got no evidence you've got something different. We found when we got samples from the Philippines in a training workshop, the PCRs didn't work and the symptoms were very obvious. So then we scratched our head and worked out we actually had a different virus present. And we had different antibodies, some worked with it and some didn't, whereas they all worked with banana bunchy top virus. It took a couple of years to get all the genome components, but yeah, it's a totally different virus. Banana bract mosaic is another aphid-transmitted virus. It occurs especially in the Philippines and in India, but there's records of it popping up in other places. There's no records of that in Indonesia at the moment. So that's a serious virus disease that you don't want to have. Banana streak virus you do have in Indonesia. That's a mealybug transmitted virus. It's a group of viruses. I think we know of 15 different viruses that are all very similar. They've got the same type of vector, they've got the same type of virus particles and genome, but their sequences are all a little bit different. That's a very interesting virus because some of the banana streak viruses, about 4 that we know of, get integrated into the genome of the banana plant. So it's like viruses in humans like HIV that integrate into people's DNA. Banana streak virus can integrate into the banana's DNA. That was the first time that had been found for a plant virus. And it just gets carried along symptomlessly and latent. And if something goes wrong, if the plant is under stress, the virus genome will get thrown out of the banana's genome, start replicating and cause an infection. And then you've got symptoms like this in the plants. We first found that when it was popping up in post-entry quarantine and all the parents were healthy and their progeny were infected. We didn't know where it came from. It came out of the genome. And cucumber mosaic, an aphid-transmitted virus, has got about 1,000 different hosts that we know of. It's probably got 10,000 hosts we don't know of. Banana is one of them. You'll have that in Indonesia. Banana mild mosaic is another one that doesn't seem to cause too many problems, but you've certainly got that here as well. So that's just a few of the viruses. And yeah, maybe 30 years ago, we only knew of 4 or 5 viruses in bananas. And we did a lot of work looking at germplasm, the International Germplasm Collection, and screening all the material. And the more and more you look, the more and more viruses you find. We've got maybe 24 viruses now in bananas. And you'll have many of them in Indonesia that you don't know are here because nobody's had time to look. Like I said, you've got these 3 in Indonesia. I think you've got banana mild mosaic as well. All the viruses can be transmitted in planting material. They have different insects that transmit them, but all of them transmitted in plant material. But by far the most important is banana bunchy top virus that we're going to be working on. Well, the next most important is banana bract mosaic virus. Okay, I think that's it. That's it. Yeah, that's the other side of our Gates project. There were 2 main parts of the project. It was looking for wild germplasm and resistance in bananas in Southeast Asia. That's where all this work here at Cultivation was done. The other part was trying to control the virus in West Africa. And this is in Nigeria, so everywhere we went, we had to have police with machine guns with us in case you get robbed and so on. But we thought that was probably a good way of controlling banana bunchy top virus in Indonesia. Luckily, the police are not friendly with us.
Yeah.
Yeah.
Thank you so much, sir.
I'll take it away.
Lilia, so
Oh, okay.
okay, before we go to the next speaker, I will give you discussion. Please raise your hand if you have any question. Students, with raise your question. Okay, 2 students. Thank you for our. See you tomorrow. Okay, Mbak.
Okay, thank you. My name is Ifaniani from Awang.
Yeah.
Sumatera.
Okay, thank you. For the banana plant, it can be attacked more than one virus in one plant or no?
Oh, yes.
Oh, yes.
And that's the same with all viruses in all plants. Just because they've got one doesn't mean they can't get another one. And especially in vegetatively propagated plants like banana, once they're infected, they stay infected with that virus. We often find that banana mild mosaic and banana street virus very commonly together, or banana black mosaic. Banana bunchy top is so severe that nobody bothers what else it has. The bunchy top will kill it, but yeah, they can have many viruses.
Okay, thank you.
Okay, the other one, are you?
Okay, thank you. I want to ask you 2 questions. The first one is, what can we do if we find the banana infected by BBTV in a large scale of plantation? Earlier you mentioned about we have to destroy the disease plants because we can't do anything when the banana is infected by the BBTV. And what if we only find a small number of infected plants inside a large scale of plantation, and how to do the eradication before the disease heavily spread, and should we eradicate the plants around it, or how? Okay, and the second question is, do the banana from the tissue culture is always free from the disease, and is it required to do the special treatment to maybe make it clean or eradicate the fibers? Thank you.
Okay. The virus is, answer the second question first. The virus is quite easily transmitted through tissue culture. You need to do something like meristem tip culture to try to eliminate the virus, because viruses move mostly, like virus like banana bunchy top virus moves in a phloem tissue, and the meristem doesn't have phloem contact right through to the tip of the meristem. So if you take the meristem tips, you can get ahead of the virus. The virus is growing up into the plant, but if you take a very young meristem, tissue culture that, you can get rid of the virus. Occasionally, just doing normal tissue culture, you'll eliminate the virus, but that doesn't happen very often. Most of the time, you'll be transmitting it very efficiently. You can use things like chemotherapy or heat therapy, but that's normally not necessary. Normally, just meristem tip culture would be enough. But you need to index the plants, because you often can't see the symptoms in tissue culture plants. All the tissue culture plants that are grown in Australia come from virus tested material. That's about half our banana production. The way that is done is that when people collect the material to put into tissue culture, they test the plants in the field at that time. So they check the mother plant. If the mother plant is free of disease, then it goes into tissue culture. That's much more efficient than testing 500 plants at the other end. Okay. Right, so that's the tissue culture part. If you only have a few plants in your plantation, the first thing is you're very lucky. So that's the perfect time to do something about it. And in Australia now we use injection guns and inject herbicide and insecticide. The insecticide is to kill the aphids so they don't move off the plant when you're killing it and infect other plants. And the herbicide will kill the plant. You might have to go back and look at it again in a month's time and make sure there's no regrowth that has happened. Also, if you do the inspections, that needs to be done fairly frequently, like every 4 weeks or so. Because if you see the symptoms very early, you can eliminate the plant before the aphid have had a chance to spread it to another infected plant. If you do quick inspections every month, and as soon as you see an infected plant, you eradicate it, then you can eliminate the disease. If you only inspect every year, and then the plant has plenty of time to, the virus has plenty of time to spread from that plant to other plants, and there'll be plants that don't have symptoms that you can't see. It takes a few weeks between infection and showing symptoms. That's why you've got to be quick all the time. That makes sense?
Okay, thank you. So more questions? Mel and Lilia from John, I'm interested in two ways of your control method. You establish a quarantine zone. How times is that? Every farmer has a quarantine zone. And the second question, you have restriction of the use of plant material. If you say that
Right. The first thing is there is legislation, there's rules, and in virtually every other country that doesn't work because people don't obey the rules. It's simple as that. In the USA, in Hawaii, they had very bad banana bunchy top virus. But just like with their gun laws, where it's their constitutional right to carry guns, it's their constitutional right to grow the bananas and not eradicate them if they don't want to. So in Africa, we've shown that if everybody collaborates in a village, the control works. If only half of them collaborate, and every second farmer's got infected plants, it just spreads back into the healthy plants all the time. So the first thing is, it's got to be, everybody's got to follow the rules, do the right thing. So the quarantine zones are legal boundaries. The whole of the area in southeast Queensland and northern New South Wales where the bunchy top occurs is a quarantine zone. You can't take banana planting material out of there anywhere else in Australia.
But there are numbers of important.
Yes, all the banana production areas, different quarantine zones. TR4 is area now is another quarantine area, a different location. And what planting material depends what it is. As I said, the tissue culture material, it comes from North Queensland where there's no banana bunchy top virus. It's all tested. It's all tested for banana bunchy top virus because someone can carry it up there and we don't know about it. We hope it's not there, but you've got to test to make sure it's not there. That material can go anywhere in Australia. But in the affected area, farmers can't take suckers off a farm that has bunchy top. They can use suckers from a clean farm, take to an infected farm, because they're putting clean material in. But there's very strict laws about where you can move planting material in the quarantine zone. Basically, you can't take material out of an infected area anywhere. Yeah. No, never happened. We tested millions of thousands of plants there, lots and lots. Yeah. Okay. Other question?
Okay, thank you. Interesting one is how does this integrated the banana genome? Is it possible to create the material pre-pilots, for example, using the physical?
No, because it's part of the banana genome.
Yes. It's interesting because we always think that virus can be free from banana bacteria with using the physical.
Yeah, not that one.
No more than
Yeah, but it's always more complicated. The banana cultivars that have got the virus in the genome are just B genome bananas. They obviously aren't a genome. There are little bits of virus in the A genomes, but they're not whole virus, just fragments. Like humans have got little bits of virus DNA in their DNA, but it's not whole virus, so it doesn't do anything. But with the B genomes, there's 4 different viruses that are in the genomes. They pop out at different frequencies depending on which virus it is. Yeah, so wherever your tissue culture, I mean, tissue culture is a stress on the plant, and that's one thing that makes the virus come out. It's actually putting the tissue culture.
So in case of integrated to the plants.
BBTV is not one of those viruses. It's only banana strict virus.
Okay. What do you think about using the tissue culture for control? And I think in Indonesia, for example, in Indonesia, when the virus already treated anywhere and in any kind of plant, it will be a host. This will be effective one or treated.
The distance the virus spreads depends on the aphid. The aphid's got to fly to the next plant. And we've done a lot of epidemiological work where you see what the percentage of infection is the further you get away from an infected plant. And by the time you get about 100 meters from an infected plant, you get very few infections. So if you were to have a plantation that had a barrier around the outside of several hundred meters, sorry, you probably limit the infection from all the sources quite effectively. So just a little bit of a barrier, a distance barrier, is very useful. If you could be a kilometre away from other plantations, it'd be very, very good. Distance is very useful.
Okay, thank you.
Oh, last question from Mas Fajar. Oh, two. Okay.
Sorry, Lilia. Listening.
They already joined.
Yeah.
Okay. Okay.
Thank you, sir. Can you hear me? Okay. Let's try to make it with the microphone. Okay, PPTP is new for me, new topic for me. Recently, some banana cultivar, well, banana, between from the PPTP system. And if you have a thought, what is the best approach to identify or study the resistance from banana? Just you know.
Okay, if time is no object, you're doing the best you can. The best thing is to aphid inoculate the plants, because if you can work out a way to avoid using aphids, like infectious clones of DNA, there's just a chance that the plants are resistant to aphid feeding, and you'll never know. So it's best to use a natural infection system, just in case plants can be resistant to that. If the virus can't get into the plant by the aphids, doesn't matter whether it's susceptible or not, the virus can't get in, it won't infect it. And you need to inoculate. The transmission is never 100% effective. So each individual aphid only has got something like a 50% chance of transmitting the virus. So you need to use lots of aphids on each plant. So you've got a 100% chance of transmitting. And then even then, occasionally a plant will escape infection. So if anything misses out in your first round, you need to re-inoculate them the second time, just to make sure that the negatives are negative. And then you need to keep them for a long time, for many months, because different cultivars can take different lengths of time before they show symptoms. Individual plants within a cultivar can vary a lot. Sometimes the plant might, in a Cavendish plant, they're very susceptible. Sometimes the plant will produce 2 leaves before it shows symptoms. Other times in the glasshouse, we've had plants that take 6 or 7 or 8 leaves before they show symptoms. So you need to keep the plants a long time. What else? I've forgotten the last thing I was going to say.
The approach to study the square system.
Yeah.
So like RNA-seq, would it be better or you tell me?
We don't know what the genetic basis of the resistance might be in the bananas. Say in the wild bananas, and some of your material that bring some of the A genomes, there might be some resistance there as well. There might be multiple sources of resistance when we eventually know, but we don't know what the genetic source of resistance is. So we've got no molecular way at this stage of screening. And any shortcuts risk losing some element of resistance. So the longest way is the most effective way of doing it, but it's slow. So it's a compromise. If you want to do it quickly, you might still find resistance, but you might lose some of your resistance sources, which you need to do the right sort of assay to find. That makes sense.
May I add one? Is there any information of the defense mechanism of the plant from this virus so far?
No, it's nothing.
Thank you.
It's a real problem.
One
One more.
Thank you for the opportunity. From your earlier presentation, you say that on bunch of symptoms only appear on the earlier stage of the plant, only on the small plant it's appear the bunchy top. But the other plant, you say that the bunchy top is not appearing, so there is no bunchy on the other plant.
Wow.
Okay, so and on the other plant there is only like a black streak symptoms or the hook symptoms that can appear. So how do you exactly that you know that the plant is infected by BBTV on the other plant? Is it always to use molecular like PCR, or you use ELISA or LP FA that you used earlier? And is it if the other plant is really infected, do you always to eradicate it immediately, or do you wait until it grow another production, or what do you do?
So the symptoms vary between different cultivars, and not every cultivar has the whole range of symptoms. You can see all the symptoms on Cavendish. Many cultivars don't have the little dark green streaks on the leaves, but they will all be stunted, and the leaves will be a bit yellow. The virus infects the phloem tissue, the phloem limited virus. That's why it's got specific aphid vector that only feeds on the phloem. So that means that the nutrients in the plant don't move very well because the phloem is infected, which is why the plants go yellow. The yellowing you'll see, the symptoms on the bunch just depends on when in the cropping cycle the infection occurs. Another thing about the symptoms is you only get symptoms on newly produced material. The virus, if you infect the plant, say it's got 6 leaves, 6 full green leaves on it, and you infect it with aphids, you might have to wait for another couple of leaves to be produced. The leaves produce one at a time in a row. So you might have 8 leaves before one is produced with symptoms. The virus doesn't go backwards. If the leaf's already been produced, the virus looks like it doesn't go backwards into the old leaves. So if the plant is grown tall, it's produced a bunch, and it's got the bracts on it, and then it gets infected, the only thing that's still growing is the bunch, which is why you only see symptoms on the bracts. So whatever stage the plant's at, it starts producing symptoms at that stage, then it goes backwards. That answer? Yeah.
Thank you, John, for very interesting lectures. Okay, before we start for the next speaker, probably I will give you 1 or 2 minutes to take the banana. Please, you can try the banana and take the drinks, probably for a little bit break. Okay, and then I will prepare for the next presentation.
Silakan, feel free. Sambil stretching.
Hey Lilia, have you joined with us?
Yeah, I'm here. Can you hear me?
Yes, I can hear you.
Okay,
Hang
I will share my screen.
Yeah, not yet. I will read your CV first, and then you can share your screen.
Can we just test and then
Okay, okay, you can test and share your screen. Oh, good. Yes, we can see your screen.
Is it working? Can you see my screen?
Yes, then the technician will explain to you first.
Yeah,
Hello?
but the sharing is working.
Yeah, the screen is working. But your screen is showing your not the slideshow mode, but the not the slideshow mode, but
You've got your notes mode.
I have my notes.
Yeah,
Okay, wait.
haven't got the slideshow.
How about now?
Yes, no, you're right.
Is it working?
Yeah, that's good. Yep, that's good.
Okay, so I will stop.
thanks for your patience.
Lilia, sorry, you have to wait a little bit because we have a short break.
Okay. And you want, okay, I will share only when you finish reading my CV. Is that right?
Yeah. Okay, everyone, if you have taken the books, please come back to the seat and we will start the next lectures shortly.
Sedang online. Sedang webinar. Jadi, itu ada omong. Mengalah lah orang webinar. Orang webinar ngomong.
Okay. Okay, I will start the next lectures very shortly. Okay, can we start right now? Okay, thank you everyone. We come back to the presentation. So this is the last speaker from Dr. Lilia Carvalhais. So Dr. Lilia is from the University of Queensland, the same research institute with me and John. It's from COFI, Queensland Alliance for Agriculture and Food Innovation. She is qualified and experienced academic with a passion for promoting sustainability, disease resistance, and resilience of managed and natural ecosystems. Her career aspiration is to use the evidence-based knowledge to achieve healthier environments. So her key strengths are passionate and engaged teaching, working with a team to manage and deliver milestones of research projects, supervising research students, writing and critically reviewing articles, reports, theses, and research proposals. Currently, she has extensive experience in managing teams and running projects, having worked on plant microbe interactions in model, wild, forestry, grain, and horticultural plant species grown in managed and natural habitats in different countries, including Brazil, Germany, Australia, USA, Costa Rica, and Papua New Guinea. Her education backgrounds, she graduated for her undergraduate in Bachelor of Science in Biological Sciences and Education, Federal University of Minas Gerais in Brazil in 2002, Master in Ecology, Conservation, and Wildlife Monitoring, Federal University of Minas Gerais in Brazil in 2004, and her PhD was obtained from the University of Hohenheim, Stuttgart, Germany in 2010. Hi Lilia, nice to see you. from Bogor.
Hi,
Okay.
how are you? Can you see me?
Yes, I can see you.
Yeah. Okay, so I'll share my screen.
Sure, you can start.
Good morning, everyone. Is it working?
Yes.
Yeah.
Yes. Yes, it works, Lilia.
Excellent. So I'll start.
Okay.
So today I'm gonna talk about banana blood disease and Fusarium wilt tropical race 4. So what is banana blood disease? So blood disease is a bacterial wilt that affects banana. So the symptoms, they include, as you can see in the photo, rotting of the fruit pulp, exudation of bacterial ooze can be often observed. There is also a red or brownish vascular staining. The leaves wilt and become necrotic. The center of origin of this disease in Indonesia, and it has been spreading in Indonesia. A very characteristic symptom is also that the male buds, they wilt and dry out, or the male flowers. And the banana bunch often looks healthy from the outside, but when you cut it, you can see the rotting of the pulp. So when the fingers are cut. So I'll just talk a little bit about the classification of this bacterium. It's part of a species complex, the Ralstonia solanacearum species complex. And this complex has been historically divided into 4 phylotypes based on molecular analysis. These phylotypes are based on 3 different genes, and they coincide with the center of origin for these different species, which were then classified later on. So phylotypes 1 and 3, they form the Ralstonia pseudosolanacearum, whose center of origin is in Asia and Africa. Phylotype 2 includes Ralstonia solanacearum stricto sensu, whose the center of origin is in America. And Ralstonia syzygiae is included in phylotype 4, and it's subdivided in 3 subspecies. Subspecies syzygiae, subspecies indonesiensis, and subspecies celebesensis, and they are originated in Indonesia, Australia, and Japan. So today I'm gonna talk about the Ralstonia syzygiae subspecies celebesensis, which is the causal agent of banana blood disease. So the data that I will present today on the distribution and transmission of blood disease were produced by Dr. Jane Ray during her PhD studies in my group at UQ at the University of Queensland, which was done in collaboration with Professor Siri Sombaidiai from the Universitas Gadjah Mada. So to determine the distribution of blood disease, she conducted a thorough literature review, as well as many surveys in Indonesia to collect samples from banana plants with symptoms of blood disease. And the pathogenicity of these isolates was confirmed using potted plants, and the identity of the isolates was determined using molecular techniques. So blood disease was first observed in Celaya Islands in 1905, and Dutch plantations lasted for about 30 years. But eventually, blood disease destroyed the plantations and were abandoned. However, the disease soon showed up in Sulawesi, and a quarantine order was put in place and was effective for over 60 years, and the disease was forgotten. However, this quarantine was lifted, and in 1987, the disease turned up in West Java. And from this point on, it has been spreading in Indonesia, now at a very fast pace. And many of these points were introduced by Jane's surveys. So I'd also like to show how this disease can spread in a single island to demonstrate its dispersal at the local level. So blood disease was first noticed on the island of Samba in 2011. And the epidemic swept across the island in about 4 years. The disease destroyed the Kepok banana industry on this island, and it's now endemic. So you can see the pattern of short distance dispersal, or the local spread that radiated rapidly from a single infection source, which is suggested that was facilitated by insects, tools, water, and contaminated soil. So during the surveys, it was confirmed that BDB blood disease was present in 154 plants, and the most common was the highly susceptible cooking banana pisang kepok. But it was also observed in a wide variety of banana across 4 species: Musa paradisiaca, Musa acuminata, Musa balbiciana, and Musa textilis. The Cavendish banana is the most popular variety, as was already mentioned, in the rest of the world, and this is also susceptible to blood disease. And there is no resistance known within the Musa genus. So Jane Ray also conducted field experiments in Indonesia. And one of the questions she tried to answer was what parts of the banana inflorescence are susceptible to infection by blood disease. So she found, through inoculation, she found that female flowers, male bell-bract scars, male bellflower cushions, snapped rachis, and the flower fruit, everything could serve as infection court. So this was based on inoculation, and it was later confirmed through molecular diagnostics. So both male and female parts are susceptible to blood disease of the inflorescence. And through these investigations, it was also apparent that blood disease is too transmissible. So for this experiment, plants were stem inoculated, and it was left to develop symptoms, and sap was used as inoculum, and 2 different isolates were used. And the disease develops in every Kepok and Cavendish plant that was inoculated with a knife. So this bacterium is definitely too transmissible. We also trying to find out if blood disease can pass from mother to sucker. And the inoculated mother plants were monitored, their suckers were monitored. And although no disease was observed after 5 weeks, all Kepok and Cavendish bananas, they had at least 1 sucker with some symptoms after 22 weeks. And in Indonesia, people commonly move banana, sucker, or beets for planting across the country. There are very little restrictions or no restrictions. So there is strong evidence to suggest that long distance dispersal was through the movement of contaminated planting material. So this life cycle summarizes how Ralstonia syzygiae subspecies celebesensis is transmitted from a diseased plant to a healthy plant. So it can be transmitted by insect and animal vectors, tools, more rarely, but still possible, through root-to-root soil and water, and long distance through the movement of suckers. The management of banana blood disease has to be done, number one, with clean planting material. So the material needs to be, for planting, needs to be assured that there is no disease. And that's the most efficient way to avoid bringing the disease to new areas. Denaveling, which is removing the male bell of the plants to prevent these infection cords that I've just mentioned. Disinfection of farm tools is extremely impossible because, as we observed in these results I've just mentioned, tools is an important transmission pathway. And also disinfection units in eradication areas or in areas that want to be left free of the disease. Although there is not a strong evidence that Ralstonia syzygiae subspecies celebesensis can persist for a very long period of time in the soil. It does persist, and it can be a pathway for infection. So this is a suggested measure that should be taken as a management strategy. So there are similar diseases caused by other members of the Ralstonia solanacearum species complex. For example, moco, which is caused by Ralstonia solanacearum stricto sensu, has very similar symptoms. It also is characterized by a typical rotting of the fruit pulp, bacterial oozing, not completely apparent from the outside of the fruit. So they are very difficult to distinguish between these two diseases. So there's also similarities in colony morphology. So, for example, although there are differences within the Ralstonia solanacearum species, some of the strains, they exhibit very close morphology. So morphology alone is not very reliable to distinguish these two diseases. So first of all, for the detection of banana blood disease, which you will do very soon in this workshop, it's very important to sample the plant material adequately. So these are some examples of plant tissues to be sampled for diagnostics. So the fruit skin, the bacteria lives in the vascular system. So any part of the plant that's not very necrotic, it's more advisable to sample from discolored material that is not very dark. It's just slightly discolored, so that there's a higher chance that there's not secondary infections in the tissue. And this is really important to obtain the cultures for follow-up with the DNA extraction. This is the typical characteristics of the colony. So in this specific media, the TZC or Kelman's tetrazolium salt, at 28 degrees, after a few days, the colonies appear with having like fluid white with irregularly round shape, and they have a pink center. It's quite easy to distinguish between avirulent and virulent strains, because when there are mutations in the media, some avirulent strains come up, and they are usually regularly round and dark red. I'm not showing this here, but I'm just mentioning. So I'd like to acknowledge a former member of my group, Dr. Vivian Ricon Flores. She led the development of the validation and development of diagnostics tools for detection of blood disease. So she validated a published assay that hadn't been thoroughly validated, which you are gonna execute soon in your workshop, which just use a pair of primers called 1-to-1, and it targets a megaplasmid, which has 1.6 mega bases inside the bacteria, it's outside the chromosomal DNA. And she also developed a high throughput specific real-time PCR method, which will not be run in this workshop, but it can be kept in mind that it's an alternative and it has been published and thoroughly validated. So for the blood disease detection, it's very important that the proper isolations of symptomatic plant material are conducted, followed by biochemical tests that can select for gram-negative oxidase-positive isolates. This is not strictly necessary, but it just helps to narrow down the number of cultures you may conduct the PCR. But this PCR alone would be able to allow you to detect the pathogen. And this 1-to-1 assay forms a clear band. It's very reproducible and robust. And if needed for confirmation, there is the real-time PCR assay called the BBD. I think it's, yeah, BBD real-time assay that was developed by Vivian. For further confirmation, it's also possible to conduct the multiplex PCR because as I mentioned in my previous slides, the Ralstonia syzygia subspecies celebesensis is affiliated to phylotype 4, and there's a multiplex that can allow you to detect at phylotype level. So I don't have much time to speak very in-depth about all the diseases, so I will move to Fusarium, Oxysporum, Cubensi, tropical race 4. I will answer the questions at the end of this talk, both about blood disease, banana blood disease, and Fusarium wilt tropical race 4. So Fusarium wilt is also a wilt disease of banana, but instead it's caused by a fungus, the Fusarium oxysporum forma specialis cubensi. It also causes yellowing of older leaves and wilting. A splitting in the base of the pseudostem can be commonly seen. Discoloration of the vascular system. I'd just like to introduce some concepts so that we understand the historical classification of Fusarium oxysporum, which is a species complex as well. So the form specialis concept derives from which specific species, plant species, the Fusarium oxysporum can infect. So for example, Fusarium oxysporum that infects tulips is called Fusarium oxysporum forma specialis tulipi. Strawberries, Fragaria, cotton, vas infection, and the banana is the Cubensi. So the form specialis indicates the strains that have the same host range in terms of the plant species. And the races, they were introduced to narrow down to the cultivar that the different strains of Fusarium oxysporum cubensi can infect. So race 1 is a race that can infect Gros Michel, Ducasse, ladyfingers, mostly banana with the genotype AAA, with the genotype group AAA. Race 2 is usually the cooking banana, like blue gold of the genotype ABB. And race 3 stopped existing because it doesn't really infect banana, only helicones. And race 4 infects all the ones that race 1 infects, for example, Gros Michel and blue gold from the genomic group AAA and ABB, and the Cavendish banana. So that's the differential. The race 4 can infect the others, while the Cavendish, while race 1 and race 2 cannot. So we'll talk a little bit about the history of and some characteristics of the race 1. So Fusarium wilt race 1, it has an Asian center of origin, so it co-evolved. It's most likely co-evolved with banana, and now it has a pan-global distribution. It's pretty much everywhere where bananas are produced. So the first reports were more noticeable when the Gros Michel plantations in Central and South America were swept out by this fungus. And the variety that the main variety of export trade was Gros Michel, so that was very devastating to the industry. So approximately 40,000 hectares of Gros Michel were abandoned or destroyed, and this strain was then termed as race 1. But the disease was fortunately controlled through the plantation of a variety that was found to be resistant, the Cavendish variety, and so the industry shifted to Cavendish banana. So the FOC disappeared as an industry problem and was forgotten as a threat. But until the 90s, when the Cavendish started succumbing to a new race, a new Fusarium wilt in the tropics in Southeast Asia. So there is the race 4 can be subdivided, was classified into subtropical race 4 and tropical race 4. So until 1889, Cavendish was only seen in subtropical regions, and it was assumed that lower temperature in this region impacted banana hosts, predisposing them to infection. However, in 1990, when Cavendish started becoming affected in tropical regions, the term subtropical race 4 was developed to differentiate from tropical incursions. However, because of the high level of virulence seen in tropical race 4 incursion, the primary research focus has been on the strains of TR4. So it was made evident that TR4 was much more aggressive and capable of infecting the previously Cavendish-resistant subgroup. And recently, a new species classification has been proposed for all the strains of subtropical race 4, which is not very genetically diverse within this race, the tropical race 4, and has been proposed as Fusarium odoratissimum. Until 2012, this strain was restricted to Southeast Asia and Northern Australia, but however, it has spread rapidly to most banana growing regions around the globe, and it's causing major losses. So the spores, they survive in the soil or alternative hosts such as weed. The spores, which can be macroconidia, microconidia, and chlamydospores, They can germinate, stimulated by root exudates, and the germ tubes penetrate in the banana roots. Then the mycelium invades the vascular systems, and the spores are constantly produced and distributed through the transpiration flow. And the mycelia and the gum blocks the xylem, and then the yellowing starts to be observed firstly in the older leaves, and as colonization continues, wilting occurs, culminating in plant death. The suckers are also affected by the disease, and they are infected through the connecting xylem of the mother plant, and the accumulation of plant debris in the soils after the plant death leads to more spores in the soil, which perpetuates the cycle. So the geographic distribution of banana plants helped with the movement of the plant material via humans and via the cultivation region around the world. So this allowed for the spread of FOC infected plant material into countries outside the pathogen center of origin, which is in Indonesia. So the early history of FOC is linked directly to the first export trade of banana. Other ways that the pathogen can spread includes infected soil on shoes, because it's well known that the chlamydospores, they can live and remain viable in the soil for decades. Clothes, machinery, tools, animals, surface, and irrigation water. But it's important to remember that the most common source of dispersal still remains the informal exchange of infected asymptomatic plant material. So we can see here how Fusarium wilt race 1 increased in the last century, or a little slightly over last century, from next to 0 countries to like over 60 countries. And Fusarium wilt, the slope of this growth is much higher. So the spread has been much more rapid, which is a consequence of the global trading, people traveling, export of plant material, exchange of plant material. So it's very important to manage tropical race 4, to have early diagnostics. However, it's also important to note that symptoms of race 1, subtropical race 4, and tropical race 4 are identical. These different races cannot be distinguished based on the symptoms in the banana host. The FOC colonies and spores are also indistinguishable. So the mycelia produces microconidea, macroconidea, and chlamydospores that are very similar, pretty much identical. So it's not reliable to resort to colony morphology or spore morphology for classification. And the classical way to diagnose FOC is through vegetative compatibility tests. These tests, they rely on the associations between the vegetative compatibility groups and races. However, these tests, they are very time-consuming. They can take a couple of months at least, and they involve the generation and characterization of NEAT mutants, which depend on the availability of tests which may not be in hand if you are in a country that has not any report of TR4 yet. So that's why molecular diagnostics comes in hand, and one of the most common tests used worldwide is a conventional PCR, which targets the intergenic spacer region and was developed by Dieter and colleagues in 2010. This test targets core genomic regions, which is within the intergenic space region, which reside on conserved chromosomes. And however, they are often present in other Fusarium species and other Fusarium oxysporum strains. And in fact, our group tested these primers and we could detect an amplicon in some strains of Fusarium oxysporum vas infectum, which infects cotton, and also endophytic Fusarium oxysporum isolated from healthy banana. And also this method was designed exclusively to detect TR4. So there was a need for a unified diagnostics for other races in banana. Often genes associated to virulence, they are considered suitable targets for diagnostics. The only putative family of effectors that have been identified so far in Fusarium oxysporum are the small proteins that are secreted in the xylem sap by the fungus when it's infecting the plant. Some of these proteins, however, they were also identified as a virulence factor in Fusarium oxysporum causing tomato wilt. So the variation of the 6-gene profile was correlated to the race structure of FOC. And these genes, they have also been detected in strains isolated not only from healthy banana, but from soil. So a very thorough look at the different polymorphisms was necessary to develop diagnostic based on these genes. The first method that used these genes as the target for a conventional PCR was developed by Fraser Smith and colleagues and reported in 2014. And that's the method used by biosecurity in Australia. This method relies on the amplification of 2 variants of the 6-8 gene, and the diagnosis of TR4 is reliant on the absence of an amplicon. And that's why we thought that's not ideal, because you can't conduct tests on robustness, repeatability, limits of detection if you are relying on the absence of an amplification product. So our group developed a series of assays that are able to detect different races of FOC. One that you are gonna run in this workshop that is based on a conventional PCR run that targets the 6-1 gene and followed up by a restriction enzyme treatment. So it needs to be seen 2 fragments as a result of a positive detection. But we also developed other assays for VCGs and races that had no reliable diagnostic assay available. So we developed 2 assays that detect VCGs 0121 and 0122 that are closely related to TR4, but don't have the same level of virulence compared to TR4. We also can detect in this assay the subtropical race 4 and the race 1. So what happens next? What do you do after diagnostics? You cannot only detect a pathogen. You have to take action. So one reason why Australia has been successful in containing TR4 is through the implementation of quarantine zones and very strict hygiene regulations in the farms, on the farms. It's very important to contain the disease because, as I mentioned, water, soil, animals, they can easily spread the disease. An example of poor containment strategies was in China. Since 1996, when there were no cases of TR4, in 1998, 14 hectares were reported, and by 2017, about 100,000 hectares, and now this must be even higher. So not having proper containment, it's very problematic to banana production in the country that is affected by TR4. A very important measure is to increase awareness about the problem. Communication with growers is extremely important. In Australia, different organizations, they run, especially the affiliated to the banana industry, they run field days. There is growers magazines where awareness about a wide range of diseases is brought to the growers. And very importantly, clean planting material as a source of planting material is extremely essential to ensure that no further spread takes place. There is a lot of studies and products that claim to manage TR4. For example, disease suppressive soils, biological control, cropping system, rice hull burning, tolerant Cavendish selection. However, a lot of these control measures or management options have not passed through rigorous field validations and studies. So to be valid, it's very important that an evidence-based approach is taken with independent tests, double-blind designs, proper controls, and that is peer-reviewed. So far, there is no 100% efficient management strategy for TR4. But resistance, as it has saved the industry before when Gros Michel was completely wiped out in the mid-1900s. There are a few breeding programs happening. For example, in the Honduras Foundation for Agricultural Research, and also soma clonal selection for example, in the Taiwan Banana Research Institute, and by the International Institute of Tropical Agriculture, which links different organizations to try to breed for resistant TR4 varieties. So far, there's a few resistant varieties, which is the FIA 01 or the gold FIA, FIA 18, and FIA 25. The problem is that in the banana, there is a range of quality standards that needs to be met, not only taste-wise, appearance-wise, but also to be able to be exported and ripe in the window that you need, because our whole chain of distribution is set up at least for export for Cavendish. Recently, regulators in Australia and New Zealand, they gave a university in Australia, in Brisbane, a group that has been working on GMO banana for over 20 years. They were granted a license to commercially release a Cavendish, genetically modified Cavendish that is resistant to TR4. However, it's still a question if how the public will accept this variety worldwide and in Australia. So I'd like to thank the former members of our team, from which I acquired a lot of the data, from whom I acquired a lot of the data that I presented today. And these are the current members of my group. Professor Andrent is the group leader, and I'm the senior research fellow. Cecilia Dwyer is our senior research assistant, and we have 2 PhD students and an honors students. And we wouldn't be able to do our research without our global and local linkages with many different organizations worldwide and in Australia. We also are very lucky to have a few organizations that have funded our work, and I'm happy to take any questions. Thank you.
For very interesting lectures, and thanks a bunch. Okay, before
I will need your I will need your help with coming close to the computer to repeat the questions, because I cannot hear very well when it's far from the computer in the audience.
Okay.
Thank you.
So before I give the chance to audience in Bogor, I will try to read the questions from online.
It's from the chat?
Yes, from the chat.
Okay. So
the first one
is one from Nofianto for me. In Indonesia, many institutional banana farming business are in the form of combinations of farmer groups or group of farmers. There are still very large companies. How to implement protection and quarantine for banana plantation, especially for fusarium wilt disease on a small farmer scale. Until now, bananas that are resistant to fusarium wilt are still being researched. Thank you. Yeah, it's tricky to have that in a small farmer scale, but I believe at least starting with clean planting material, it's a good start. Sourcing the planting material from a reliable provider. Having hygiene, procedures implemented locally, for example, even a cleaning of farm tools, preventing people from outside walking with dirty boots in the property. So far, it's the research in terms of finding resistant varieties and developing resistant varieties is still lagging behind compared to other crops. But I think, yeah, a good start is having like a basic, farm hygiene and clean planting material from reliable sources.
Thank you, Lilia. So actually, there are 3 previous questions for the same person. The first one, what gene causes kapok and Cavendish bananas to be susceptible to blood disease? And how does the micro...
Which genes? Say again.
Yes. What gene causes kapok encephalitis bananas to be susceptible to blood disease?
This is not known, the genes that are affected to susceptibility, because even the resistance of different because resistance is unknown, it's difficult to assess susceptibility, because pretty much as far as we know, all the varieties that have been assessed so far in Indonesia, including Cavendish, they are susceptible. So I don't think that this knowledge is available yet. The genes in the host that are linked to susceptibility. I think that would need to be done through like a genotyping of banana germplasm, which, as far as I know, have not been done yet.
Okay.
At least for blood disease.
Okay, thank you, Lilia. The second question was, how does the microclimate influence the effect of blood disease and the influence of climate changes?
Are these questions for me too?
Yes.
Is it in the chat?
Yeah.
Okay. I can't see the questions in the chat because I find it hard to understand even you. Okay, genes. Now I can see. How does the microclimate influence the attack of blood disease and the influence of? Well, that's a good question. Microclimate in terms of, I mean, as far as we know, blood disease is in tropical. It has only been found in the Philippines and Indonesia. So I think microclimate, I'm not sure if that has been done yet, or what exactly it means. I mean, we experiments on like for example, humidity and other abiotics factors, for example, pH or salinity, drought, has not yet been assessed for in terms of the virulence of Ralstonia syzygia and celebesensis. And climate change, it's also, as far as I know, I'm not sure if there's no study that affects the influence of climate change. We know that with climate change, there will be more unpredictable extreme weather events, and these events may cause more proliferation of vectors, insects. Who knows? But we know that for blood disease, there is not a very specific factor. It's more like the sap is transmitted through birds, bats, and climate change influencing the behavior of animals and of different insects may influence the spread of blood disease. That's a bit hard to model or predict at the moment. So many people plant bananas in rice fields or yards. Does this also affect the incidence of important banana disease, such as Fusarium wilt, banana bunch top, and blood disease? Well, yeah, rice fields, as far as I know, are grown in flooded areas often, not everywhere. But water is a very well-known transmission pathway especially for Fusarium TR4, like flood water and irrigation water. So I think, you know, having water can affect the spread of the disease. For bunchy top virus, I cannot. John would be more knowledgeable to answer this question. I wouldn't know how to answer. And with blood disease, for the rice fields, I'm not too sure. Like blood disease is not as prolifically spread by water and soil compared to Fusarium or even Moco, the other disease called by Ralstonia solanacearum stricto sensu. So I would think that TFO would be most likely more affected with, you know, depending on what you plant together. But in a way, like having more diversity in your field prevents the spread of disease. So the problem with Gros Michel and Cavendish, Gros Michel back in the 1950s and Cavendish in Australia is the lack of diversity. Is only one variety planted in an enormous areas. This is like it's just so conducive for spreading of the pathogen. But when you have a more diverse cropping system, such as in Indonesia, it's much harder for the disease to spread at the same rate compared to monocultures that only use one variety. So this is well known from, you know, cropping systems that when you have cropping of multiple plant species and trying to have like fallow periods and also, you know, not plant, although with banana is a bit hard, no? It's more of a perennial crop. But I think increasing the diversity in the cropping area of varieties and plant crops is definitely a way of preventing very prolific spread of a pathogen. That there is no doubt about that. I think that's why in Indonesia it is still possible to produce banana despite all the banana diseases that are in Indonesia. It is still for example, in the case of blood disease is a little more complicated because, you know, people still have considerable areas that they want to commercialize the fruit. And if there's no resistant varieties, it will be very hard to produce something that can be, you know, eaten and commercialized. But having more diversity is definitely advantageous comparing to extensive monocultures.
Okay, thank you, Lilia. Moreover, I also would like to invite the Dr. Audrey from Patimura University. I will give her to us. Lilia, have a question? Is Dr. Audrey there? Oh, here. Oh, I think she's somewhere. Okay, I will give time for the participants in this room. Is there any question? One from Mbak Fitri, two from Mbak Amel, three Mbak Henry. Okay, Lilia, you have further three questions from the participants on site. Okay, I will give to.
So you have to help me to read the questions close to the computer.
Oh no, they will ask you directly.
Yeah, okay. I'll try to listen. I'm just saying if I can't understand, I may need your help.
Okay, thank you.
Okay, thank you. Lilia, can you hear me?
Yes.
I'm Fitri from Indonesian Quarantine Agency, and I'm very interested with your presentation, especially about Fusarium.
Mm.
And Fusarium is our concern in quarantine, especially in oil palm, that is Fusarium oxysporum forma specialis elidis. That we know that Fusarium is very complicated and very confusing to identify morphologically in our work. We have experience to develop methods about Fusarium oxysporum elidis using molecular, and then we have a problem to grow and to distinguish morphologically in PDA medium. Based on your work, maybe you have any suggestion, because sometimes we very difficult to test this is false or not based on morphologically characteristic. Because in PDA medium we it's too hard to find macromolecular in PDA and very rare to find in PDA medium. Maybe you have any suggestion for us. Thank you.
Well, as I mentioned in my talk, the Fusarium oxysporum, the different forma specialis, they are very hard to distinguish morphologically. The culture morphology and the spores can be very similar. They are very similar usually. So I wouldn't try to distinguish based on colony morphology. I think the best approach would be to find specific regions in the Fusarium oxysporum that you are working with. I think you mentioned the palm oil, Fusarium oxysporum. And this can be found based on you sequencing or trying to run fingerprinting. That was the old school method, but right now, since the cost of sequencing genomes has decreased significantly in the last decade or so, I think, trying to find specific regions in the genome for your forma special and trying to design methods, and most importantly, validate these methods really well based on specificity, so include closely associated species and also other Fusarium that are known to infect banana. Also, a lot of Fusarium oxysporum are just endophytes, includes those as well, and running a proper validation. But definitely doing diagnostics based on morphology is tricky, and that's why molecular diagnostics can be really useful for species that have can be so similar morphologically.
Hi, Lilia.
Hello.
Hi, my name is Amelia. I'm from university in the southern part of Papua. Yeah, so it's close to the Christmas Island, I think, yeah. It's close to one of the Christmas Island. My question would be about the banana blood disease. You said that there's ones that not virulent and there are ones that virulent. And you distinguish that based on the colony, if I'm not mistaken?
Yeah, in this medium, it's common that some of the colonies they go through mutations and they become avirulent as a consequence of multiplications in the media. But
Okay.
they can be easily distinguished, the avirulent and the virulent, in this particular media that I mentioned.
Oh, okay, okay. So is that already published?
Yeah, there is a lot. Yeah, there's quite a few studies that show that. But these avirulent, they are not like, they are just a consequence of growing the bacteria in the medium. They suffer because the bacteria they multiply so prolifically in the medium. Some of them will suffer mutations and they will become avirulent. But then the idea is that because for our protocol, it's based on DNA extracted from isolated colonies and not from plant material. So it's important to choose the right colony in the medium. That's why I mentioned that sometimes there are colonies with atypical color, texture, and sizes and shapes that can be easily distinguished in the media, that you shouldn't take those forward because they will be avirulent.
Okay, okay, thank you, Lilia.
Thank you. Next question. Yes.
Okay. Hello, Lilia. Can you hear me clearly?
Yes.
Yeah, okay. Great. My name is Henny, and I'm from Nusa Tenggara University in West Timor. I noticed that on your acknowledgement, there's also the name of my university. So actually, at that time, there was two of my colleagues working together with Prof. Andre Grant and also Jane Ray for doing sampling in Sumba and also Flores probably. And yes, following that result and also the recent situation that is also happening in West Timor and especially in Flores and also in Sumba, we were experiencing like an outbreak of the banana blood disease actually. So previously in the report by Jane Ray et al., Flores has not yet been infected by the banana blood disease, but now all the island from corner to corner is already wiped out of the banana blood disease. And we were using also, including me and my students, also checking about the samples coming out from Flores using the primers that you were providing, the one-to-one for 100 readings, and then all detected positive on that.
Yeah, they were they
They
tested
were
positive.
Yes.
Yeah, because I remember Andre and Jane, they went to Flores and they couldn't find diseased plants. So now it has been more predominant.
More, yeah. But we were hoping another island in Timor will not be infected by that bacteria.
Yeah, make sure
Yeah,
you
that's
are
a
careful with
pity
it.
actually, yes, bad situation. But I want to know whether, so since you were using the one-to-one that is comes out from the plasmid bacteria, and then that is only producing around 317 base pairs. Do you think, is there any specific, other specific markers that can be used? Because now it seems to be that probably there will be diversity already of those bacteria in the surrounding and then the infection. Indeed, mostly what is infected is the kapok, also the one that you found, but we also saw it and also in Barangan and also Pisang Susu.
also we found in that. So I was wondering whether there is other marker that can be used instead of one-to-one marker. And then in terms of Fusarium wilt, the tropical raceworm. So
Can I
you
just reply this one before you move to?
Sure.
to
Yeah,
to
go ahead. Yeah.
So yeah, that's interesting that you are saying that now it's more predominant in Flores, because from the surveys that were conducted, I don't think they could confirm that the disease was in Flores. So that's a good update. You should definitely try to report that in a publication. And from our observations, there is not that much diversity. The population of blood disease in Indonesia is still reasonably clonal. There is not that many markers differentiating different. There's not that many strains of blood disease, to put it that way. So we have not observed any false negatives with the one-to-one primers so far. That doesn't mean that, you know, that a mutation or some, you know, transposition event can happen, and the particular regions that in the megaplasmid that the primers target will be affected. But we did publish an alternative assay that's based on another platform, the real-time PCR, that can be used as a conventional, as a alternative method. There are other methods available, but so far we have not considered them as reliable. I think first it would be important to have a study of the genetic diversity of the isolates that are spreading now, to see if they are very different from the ones that we've used that were used with the one-to-one primers were not developed by our group. It was developed by Mark Fagan a long time ago. So I think it's 2004, these primers, and they are very robust. But I think a genetic study evaluating the diversity is firstly needed to then assess the necessity of developing a new molecular diagnostic method. So I think it's always important to keep collecting material, keep recovering isolates, and determining if the phenotype or the behavior in terms of infection, symptomatology, host range is changing. That would be reflective of the genetic diversity. And also keep trying this. I don't think new methods needs to be described. Of course, alternative methods are important. But you know, if you are saying that the one-to-one primers, they can detect the isolates that you have been recovering recently, although I think it's extremely important that it's the assessment of the genetic diversity is relevant to find and develop new markers. So far, it's not needed if you see that this is working for you right now.
Yeah. Okay. Yeah. I was just wondering whether has ever been the work for like doing the whole genome sequence from that plasmid. Yeah, or not.
Yeah, there are genomes of banana blood disease available on NCBI that can be, yeah. And the megaplasmid is part of the genome, is an important part of the genome. So it will be there too.
Yeah.
But it doesn't need to be limited to the megaplasmid, you know, because
we can take it from anywhere, you know.
Yeah,
right?
Yeah, anywhere that it's specific to the group of blood disease that you may missing out with the current methods if they are no longer reliable to detect.
Yeah, I think, yeah, but so far 1-to-1 is still reliable, still really reliable actually, just curious. Can I have another more question?
Yeah, I don't know. I mean,
One short, okay. Related with the tropical rainfall, you were mentioning also there are several grouping about subtropical, tropical rainfall, and also odoratissimo. Have you been able to differentiate using the 6 markers or 6 genes to differentiate whether they are FOC and also from other Fusarium ovisporum, and whether you were able to differentiate also between the subtropical, tropical rainfall, and also odoratissimo.
well, as I mentioned, the Fusarium odoratissimum is a synonym to TR4. It's the new species proposed for TR4. It has been proposed by Mariani and colleagues, I think in 2019 or more recently. Yeah, it hasn't been widely adopted yet, but the proposal is there. And there is some evidence to support that it's a different species. And as I mentioned in my presentation we are able to, of course, some of the assays, if you look at our publication, there will be some limitations, but in general, for TR4 is quite reliable. So, and also we can detect subtropical race 4 and most of the strains of race 1. Race 1 is quite diverse. So some of the isolates we were not to detect. We were not able to detect. But you mentioned about detecting other former specialists. So there is another study that I don't think I mentioned here from Skilovsky and colleagues from the University of Queensland as well, where they characterized the different alleles of the different 6 genes within the races. And they found that the 6-9 gene is usually present in all the races of Fusarium oxysporum cubensis. So this could be used as a marker for Fusarium oxysporum cubensis. And in our assessments for our conventional PCR assays, we have included, if you see in our publication, we have included isolates from many different former specialists, and we were not able to detect with these tools different former specialists that are not cubensis. So the aim of our study was not to have a tool to distinguish former specialists, but was to have a tool to specifically detect TR4 subtropical race 4, race 1, and these 2 VCG0121 and VCG0122 that are closely related with tropical race 4, but they are not as virulent as tropical race 4, although they can still infect Cavendish, but they belong to a different cluster in the phylogenetic analysis. So they are not TR4.
Okay, all right. Thank you very much for your answer.
Thank you.
Thank you so much, Lilia. Yes, when we are talking about the banana disease, it looks like never-ending interesting topics. Okay, because it's time for lunch, and I know it's afternoon already in Brisbane.
Yeah,
So I
3:20.
would like to say thank you. And before we end the guest lecture, I would like to give a certificate So probably the first one will be to Dr. Lilia. So I will give you the certificate through online, Lilia. So
Thank you.
the printed version, Songa and John will bring it for you.
Great. Thank you.
So this is to certify that Dr. Lilia Carvalhais as a speaker has joined this lecture on pathogen diagnostic on 25th of June 2024. Thank you so much for your lectures. It's very interesting.
Thank you very much. It was a pleasure to talk to you all, and I'm very pleased that there was so many questions and interest in that talk. That not everyone was sleeping.
Now we have a coffee and banana as our snacks. Okay, the second one will be Professor Awang. Unfortunately, he has left. So thank you, Professor Awang. We say thank you through Zoom. Already delivered about banana in Indonesia. And for John, probably Bu Asti will give it directly with John, please. For this one.
Okay. Okay, John, thank you very much for very nice lectures. It's very informative and bring us the better knowledge and understanding about banana bunchy virus. Thanks. Thank you very much.
Thank you so much, and thank you so much for our participants through Zoom. I know there are still remaining questions. We will deliver the questions through the speakers, and they will try to answer directly to you. And for all of the attendees, an e-certificate can be accessed to this link. Okay. And thank you so much and have a good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you.
Thank you, Lilia.
Enjoy your
Bye.
time there.
Bye.
I wish I was there. Bye-bye.
Yes. Bye-bye. Baik, Bapak Ibu, terima kasih atas kehadirannya untuk peserta workshop yang khusus peserta workshop boleh tetap di sini. Nanti setelah makan siang akan ada sedikit briefing terkait field trip besok. Terima kasih.
Lagi nggak, Mas?
Ya.
Foto yang di luar, nggak?
Boleh mau sekarang?
Baik Bapak Ibu, untuk kita akan dokumentasi sesi kedua karena tadi ada beberapa yang belum hadir. Kita akan take a picture in the outside before we have lunch. Mari ke tangga ya, seperti biasanya. Mari Bapak Ibu ke tangga.